microscopy room at UGA. My name's Annie. I'm a second-year PhD student in the Institute of Plant Breeding, Genetics, and Genomics, and my research involves using nuclei and RNA-seq data sets to try to understand how plants grow and develop and produce food, biofuels, and even medicines. And we use nuclei for a lot of different reasons, not only for RNA sequencing, but also to estimate a plant's genome size and also to count chromosomes to see what the plant's ploidy level is. In my lab, we study a ton of different plant species and isolate nuclei from diverse tissue types, including tubers from potatoes, leaves, stems, roots. And so we've had to optimize this protocol to work on a variety of different tissue types and different species. Here I'm using fluorescent microscopy to visualize the nuclei that I've isolated. I use a DAPI stain to stain the DNA, and then I can visualize the blue dots under the microscope. And a good quality nuclei will be nice and round without any visible signs of damage, whereas a bad quality nuclei might have mRNA or chromatin leaking out the sides. And you can visualize this and then get a ratio of good nuclei to bad nuclei to decide whether or not the protocol worked or whether you need to optimize it more. To make the nuclei buffer, this is the recipe you're going to want to follow. You can make this in any final volume, however, you're going to need at least 2 milliliters for every sample. Use water to bring the buffer to the final volume that you need. These are the reagents that were listed in the recipe slide. You can pause to take a closer look at any of the brands that I've been using. But you could also swap some of these out with whatever your lab has on hand, as long as the final concentrations are the same as in the recipe. And finally, DAPI is used to stain the nuclei for viewing and for flow cytometry. This is a list of all the tools you're going to need, and this is what they look like. The glass petri dish and razor blade are used to chop up the tissue to release the nuclei, and the filters are used to remove the cell debris. Do everything over ice to preserve nuclei integrity. Go ahead and make the buffer, then chill it over ice. Here, we're using 0.025% Triton in a 2 milliliter aliquot for one sample. You can use either frozen tissue or fresh tissue. Weigh out 0.1 to 0.5 grams of tissue, then add 1 milliliter of your Triton buffer. Once you've added your Triton buffer, rapidly chop the tissue with a razor blade for about 2 minutes until it's the consistency of pesto. You can scrape the tissue so that all of it is submerged inside the buffer. That way, when you break open the cell walls, the nuclei will be released into the buffer. The physical chopping helps break open the cell walls, and then the Triton will lyse the cell membranes to release nuclei. Add the second milliliter of Triton buffer, then rinse the tissue a few times to make sure as many nuclei as possible are collected. Use a pipette to pass the nuclei through the various filters. I like to pre-wet the filters before doing this to make it easier to pass through. If you're having trouble making the nuclei buffer flow through the filters, you can tap or spin to make it go through faster. And that's it, your nuclei are ready. You can take an aliquot of the liquid and spike it with DAPI to visualize. Our DAPI is at a concentration of 1 milligram per milliliter, and we usually just add 1.25 microliters into our aliquots. You can mix up the nuclei gently by inverting it a few times. Don't forget to put it on ice so that the nuclei will last longer. Here I'm taking it to the flow cytometry facility in order to purify them for single-cell RNA sequencing. To learn how to do flow cytometry, check out Trent McCracken's video called Using Flow Cytometry to Identify Ploidy Level in Holly. He walks you through how to use and interpret flow cytometry. Here I am evaluating the nuclei to see if the yield and quality is good enough for single-cell RNA sequencing. Hope you guys learned something. Don't forget to leave a comment down below in case you need clarification on anything. Also, there's a link inside the description of this video that describes how to use flow cytometry, and also a link to the publication that goes over this method in greater detail. Be sure to check those out. Thanks for watching.